has many published scientific publication in various reputable journal, and has many international patent about fluorescent probe for lifestyle imaging and many awards. Okay, so today Ibu Samira Husen Alamudi Ph.D. will be sharing valuable insight on physicochemical properties of drug molecule. So please everyone pay close attention and please actively engage by asking question. Okay. Now without further ado, please welcome Ibu Samira Husen Alamudi.
Thank you so much, Dr. Isna. Hi, good morning, everyone. Give me a second. Let me share my screen. All right. Can anybody see my screen?
Yes, doctor, we can see.
Okay. Okay. So I'm assuming my voice also loud and clear. Yeah.
Yes, doctor.
Thank you. Okay. Hi. A very good morning to everyone. It's nice to see you again. Some of you, I see quite a number of people here. Maybe more people are joining us. So today we will continue our lecture from last meeting, the first lecture by Professor Zahir Jude. So last week we have learned about the introduction of drug design and discovery. So today we will continue from that. We will talk about what are the properties that needs to be considered when you designing a drug molecule, right? Okay, before we start, I want to have some, I want to gauge the level of expertise from all the participants here, because I may not know every one of you. So I just want to know, like, in what year you are currently at. So all of you, can you please go ahead? If you have the laptop, you can just click menti.com and key in the passcode, or you have your mobile phone with you, you can just scan this QR code. There is one quick question. Could anybody just scan and then answer that question? If you have difficulty in scanning the code or getting the access to menti.com, please let me know. Let me just go to Menti and see. All right, I see some of you have already answered the question, but I'm still waiting for more of you. We have about almost 30 people here, and so far I only got like 14, 15. So, all right, can anybody? Okay, more people coming. So here, the passcode for those who just joined and haven't answered the question. More people are coming. So just a very quick question about what level are you currently at? Right. Okay, so let me just see. Okay, how to share this. Okay. Okay, so I see, okay, many of you are bachelor degree, most of you at year 3 and year 2. Okay, we have some master students. Okay, okay, good. So the majority of you are undergraduate students. Okay, nice. Thank you, everyone. Okay, nice. Okay, let me just reshare my screen again. Okay, great. Thank you, everyone, for answering that question. So for this lecture, this is the recommended books that I suggest you to have further reading if you're interested in medicinal chemistry. So I have these two titles: The Introduction to Medicinal Chemistry by Graham Patrick, or you can also read The Medicinal Chemistry by Thomas and David Williams. I think these are available online. So it's like the soft copy or the PDF of the book, you can get it easily online. So that's why I suggest you to have further reading on this. It's a very comprehensive book if you are interested in this field. Okay, so today we will talk about the introduction of drug likeness. As I mentioned before, last week we have learned about the introduction of what is drug design, what is the drug discovery, and now we want to talk about what are the desirable properties of the drug so that it can go, or it can be called drug molecules, not only the candidate, but drug molecules. So that kind of property is called the drug likeness. And then after that, we will talk about some of the key properties to more details, such as the ionization, lipophilicity, and solubility. And I hope that this session will be more interactive. So that's why during the lecture, we will do some little practice and discussion as well. Okay, so we have learned from last week that the majority of drugs fails after the clinical trials, right? If we see the rate is about more than 90% of failure rate. Why is that so? Now, so the drug discovery and development is actually a very, very long and costly and high risk process. The time it takes from the beginning until the drug can be approved for the market is about 10 to 15 years, if you are lucky, right? 15 years. And for the NME is the new molecular entity, the new drug molecule, basically. The average cost is about 2.5 billions of USD. So it's a very costly, all right? And if one candidate of drugs is successfully go to pass phase 1 clinical trials, it's already considered as a big achievement for the pharmaceutical industry or those in pharmaceutical field, because it means that the drugs have been rigorously optimized at the clinical stage. So however, still those drugs, when they go to clinical trials, phase 1, 2, and 3, majority of them also fail, right? Even at the very last step, the drug approval, as Prof. Zahir mentioned before, it has chance to also fail at that stage, right? So if you counted all the steps, it's actually more than 90%, is a very high failure rate. So what are the possible reasons for that to happen? So if we can see, there are 4 at least possible reasons for the high failure rate. First, the lack of clinical efficacy. Clinical efficacy, it means just the drug simply didn't work. So it's just failed. It has no effect at all to the body. So lack of clinical efficacy. And then the next possible reason is the unmanageable toxicity. We need to consider whether the drug may have the side effect to your body, right? The negative effect, so it's unmanageable toxicity. And then the poor drug-like properties. For example, some of drugs are designed for the oral administration, some for the IV and all that. And the poor drug-like properties, it can lead to the drugs being ineffective when administered to your body, right? And then the last reasons about the lack of commercial needs and the poor strategic planning. So if you see the whole thing, so actually is like less than 10% of successful rate, even like 5 or less than that. So here I want to refresh you about the classical process in drug development before we move further. So I think you have seen this kind of processes before. So that the drug development process started from target validation. Let me just on my pointer. Okay. From the target validation all the way to approval to launch, it takes up to 15 years, right? So if you see here, during the target validation, it includes when you need to design the disease model, you do the target identification, and finally the target validation. It takes 1 to 2 years. Then after that, you can move to the compound screening here. The compound screening, if you see here, the number is more than 10,000 of compounds candidates to be screened at the same time during the screening, right? Because of this huge number of compounds. So the method that usually be employed is called the HTS, high throughput screening. From these 10,000 of compounds, you screen through the targets in vitro. Maybe you are lucky, you get few hundred of candidates that can pass through that stage and then move to the lead optimization stage. So during this lead optimization, usually it also together or coupled with the preclinical test, because you can do the testing not only in cells, but also move to the animals. So usually the lead optimization and the preclinical, they go hand by hand. During this stage, usually we can optimize the drugs from the compound screening. So you can do kind of reformulation. So you can adjust the drug-like properties, the solubility, the permeability, and then you can also do the SAR, the structure activity relationship analysis and all that. Then you can also predict the toxicity of the drugs in the preclinical test. From here, you may get, if you're lucky, less than 10 candidates out of more than 10,000 candidates. Then you're happy, and then you move forward to the clinical trials, right? The clinical trials, then you can also check how the drugs behave in your body. You can adjust the dose, and you can check the toxicity and the efficacy. And then if you are really lucky, then we can get 2 or even 1 successful candidate that we can call it a drug. So the whole process is really, really long process, and is very costly, right? So what makes a successful small drug molecule? So there are many factors to be considered, but we can see 4 major factors here. First is the pharmacodynamics. It is actually whether the drug works, how the drug works at the target of your protein or whatever the biomolecule target in your body. Or in other words, it is a measure to check the compound ability to interact with the intended target, not with the other target, but the intended target. So we talk about the potency, the activity of the drug, and also whether the efficacious of the drug. After that, another one is so-called pharmacokinetics. It talks about how the drug gets to the target. So you talk about how the action of the body on the drug. It's about the ADME, we call it ADME, administration, distribution, metabolism, and exertion. So these 2 will be, I think we will discuss further in the later lecture, I think the next week or the next 2 weeks. And then after these 2, we have developability. It's a funny word, right? Developability. So it talks about the chemical synthesis of the drug, the formulation, and the stability. And then the next one is the toxicity, the safety of the drug. We hope the drugs to have no side effects, right? So how, as a chemist, right, how we can adjust this whole thing? The pharmacodynamics is about, we cannot really modify much about this, but for the drug likeliness, we can adjust from the point of view of pharmacokinetics and the toxicity. So these 2 we can play around with, because these structural properties can produce the acceptable properties, the level of pharmacokinetics and toxicity. So that's why now we will talk more about this drug likeness. Right. So the poor drug-like properties, I call it, not I, we call it drug-like, because the compounds may or may not be drug, right? So we call it drug-like, the candidate of drug. So last time in the 1990s, usually the poor drug-like properties, it contributed to 30 to 40% of value in the drug development. But these days, the value rate is decreased. Now it's about 10 to 15%. So we're happy, right? So this improvement benefits from the very rigorous selection criteria when we do the designing and we consider the drug-like properties. It includes the solubility, permeability, the target binding interaction, and the metabolic stability, and also the bioavailability for the case of the oral drugs, right? So the pre-formulation and formulation studies are really conducted to increase this kind of properties, the drug solubility, and hence can improve the oral bioavailability. So we will talk about this later, the rule of 5 that have been considered in the chemical structure design. This is for later. So here, among all of these properties that you see, the drug discovery is actually a multi-parameter optimization. You see the ionization, solubility, permeability, lipophilicity, selectivity, potency, and everything. All the parameters of interest are usually interconnected. So what happens if you try to adjust one parameter? Let's say you adjust the solubility. You want to increase the water solubility of one compound. It's not necessarily only to increase the solubility, but it may affect the other parameters, such as lipophilicity, right? You increase the water solubility, the lipophilicity will be decreased. So as a chemist or as a designer, I would say as an architect of designing the drug, you need to try to balance when you adjust this kind of properties, because most of them are interconnected. So the drug-like, as I mentioned before. So if we can make a good narration of it, is a compound that have sufficiently acceptable ADME properties and toxicity properties. And we hope that it can survive through the at least phase 1 clinical trials, right? So these drug-like properties confer good level of ADME and toxicity characteristics. And this actually an integral part of the drug discovery field. So for this kind of properties, as a chemist, we can control through the structure modification. For example, you want to increase the solubility. So you can add more, the charged molecule, for example, and other things. And for biologists can use these properties to optimize what kind of bioassays that they need to perform and how to interpret the biological experiments. So here, the structure of the compounds actually determines the compound's property. For example, here, when the structural properties interact with the physical environment, it can cause a thing that we call the physicochemical properties, for example, solubilities. When I talk about structural properties, for example, these are like molecular weight, the number of hydrogen bondings, the number of the charge functional group, the lipophilicity, the reactivity, like how is the surface area charged, something like that, right? And when these structural properties interact with the protein as a target inside your body, they can cause what we call the biochemical properties, such as the metabolism, the absorption, kind of things. At the more level, the higher level, when these two properties, the physicochemical properties and the biochemical properties, interact with the living system in our body, they can cause what we call it the pharmacokinetics, the PK, and the toxicity. And we as a chemist, I would assume we as a chemist, right? So the chemist can control this profile, the toxicity profile, the pharmacokinetics profile by modifying the compound structure. So here, the hierarchy actually. So from the structure, we can get some properties, and then the properties can lead to a certain profile. So we can modify the structure to obtain the desired target property profile. So you can see here, the structure modification. I hope you can see my pointer, yeah? The molecular properties. You can play around with the size of your compound, with the shape of the compound, the number of the rotatable bonds, because it also reflects the flexibility of your compound, the polarity, lipophilicity, and even the ionization form of your compound. This will affect the physicochemical properties and biochemical properties, like chemical stability, solubility, permeability, and others. And this can lead to the pharmacokinetics and toxicity. or we call it the profile of the drug candidates. So this is just one example. I think for you, this is a very common structure. If you see here is a thymidine, right? Thymidine is one of the building blocks of your DNA, nucleic acid. Nucleic acid, we have 4 building blocks. A, C, G, T. This is the T, thymidine, right? Thymidine, naturally, it's inside your body, right? It's important to make the nucleic acid inside your nucleus, right? But what happened here? If I want to play around, I change one functional group of this. Here you have the hydroxyl, right? OH. I change into this very small functional group. It's called azide, the N3 azide. And the rest of it, I don't change anything, right? So the small structural modification, it actually can lead to quite a big change in the properties. So you see here, this is actually a drug. It's already a drug. It's called the AZT, azithromycin, or we call it the zidofudin. It's actually the anti-HIV. It's antiviral drug. So you see a very small change, hydroxyl to the azide, is already changed the whole properties of the drug. And this is what actually already marketed. So you can find in the market to treat the HIV disease. Apparently, this AZT is a very selective and it's very potent to target one protein called the reverse transcriptase, right? Actually, I want you to have a look at this structure, right? You know, the thymidine is actually the building block of the nucleic acid. Why you think when you change this hydroxyl functional group at the 3' C carbon here, the properties change drastically? Anybody wants to share their opinion?
Does it have something to do with hydrogen bonding by any chance?
Again?
Any difference, change in hydrogen bonding,
Yes,
possibly?
correct. Yes. So this can make the hydrogen bonding, right? And now no more. But what is the implication of that? If you think about it, the nucleic acid, right, is the polymerization from one building block, one to each other, and then they make like a very long strings of nucleic acids. This actually is responsible to interact with the other building blocks. When no more hydroxyl here, what happened? It stops making the strands, right? So that's what happened here. And apparently it's very selective to specific target protein, reverse transcriptase. So this is how you can do some structure modification. It looks simple, but we know it's not that simple. But this is how you can make a drug. Any questions so far? If you have any question, you can just raise your hand or you can just unmute yourself, okay? Any questions so far? No? Okay, good. So we comes to this, we talk about these physicochemical properties a lot, right? So physicochemical is a fusion of the two words, physical and chemical, means physical and chemical, of course. So these both of the properties can invoke the pharmacological response to the target or the receptor, which can be biological molecule or any system with which it interacts, because the drugs, for it to be potent, it has to be interacted with the target inside the body. So some of the key properties that we will talk more in details are 3 of them: ionization, lipophilicity, and solubility. Let's see one by one. Okay, first is the ionization. So what is ionization? Is actually a process, right, in which an atom or a molecule can acquire a positive or negative charge by losing or gaining electrons. That's what we have learned at year 1. So hopefully all of you already learned this, right? Because I don't see any of you from year 1. So most drugs are actually designed to be weakly acidic or weakly basic substances, and then because of that, hence it can be ionized at the physiological pH. So why is that so? Because if we talk about these drugs, right, we need to talk about the drug need to be absorbed by our cell, need to pass through our cell membrane. So the passive diffusion across the lipophilic membrane, it depends actually on the degree of the ionization. Why is that so? Let's look at this small figure I have here. So you see this, some of the small gases like oxygen, carbon dioxide, and everything. It can pass through easily to our cell membrane. Let's say this is our cell membrane, right? The lipid bilayer membrane. It can cross this very easily. And we can move to a bit bigger compound like benzene here. Benzene is a very hydrophobic compound. It also apparently can move easily to pass through the membrane. And the same thing about the small polar molecule like water or ethanol. It can also be easily passed through the cell membrane. But what happened to a bit larger molecules, polar molecules like glucose? Apparently, glucose cannot be readily absorbed by the cell membrane. So that's why for glucose, you have this glucose transporter to transport the glucose from the extracellular to intracellular. How about the other molecule, like this charged molecule, like this sodium, this chlorine and everything? It's a very small molecule, but it's a charged molecule. Apparently, this cannot pass through the cell membrane. Why is that so, you think? If you consider the size is very small compared to, say, ethanol or benzene. But apparently, it cannot be readily passed through the cell membrane. Anybody wants to give opinion?
Can I have
The
Yes. Okay. Maybe one by one. I cannot see the name of you, so yeah. Please,
I
please
guess I'll
unmute.
start first, okay? Is it all
Yes.
right?
Yeah, sure, sure. Go ahead.
In my opinion, I think it's because the charged molecules, or as well as those metal ions, as well as the hydrogen ions, have different polarity with the phospholipid bilayer.
Yes, correct. Good. I see some. Anybody else? I see Adam. Is it?
no, mine was going to be the same due to the fact that it can't interact with
Correct.
the non-polar fatty chain layer in the
Exactly.
lipid bilayer. Yeah.
Yeah, because in the lipid bilayer, you see it has two what do you call characteristic, right? On the surface is actually is very charged, right? Because of the phosphate, right? In the middle is very hydrophobic. So this actually will be it has the repulsion from the charged molecule. So even though the size is small, but the charge itself prevent it from coming easily or pass through the cell membrane. So from here we can move. So the ionized drugs are usually they are hydrophilic drugs and thus cannot cross the cell membrane easily. In contrast, the non-ionized drugs, they appear to be more lipophilic, and this they can be easier to penetrate the cell membrane by simple diffusion. And why is that important, the ionization? Because it has major effect on many properties and parameters. So when we talk about the ionization, I want to bring your attention to this pKa, because most of the drugs I mentioned before, mostly weakly acidic or weakly basic. So we will talk about this pKa more further, yeah? So the number of the pKa, the value of pKa, it will reflect to the different properties. The permeability will be affected, the solubility will be affected, and the interaction with the cell surface also will be affected. So most of the drugs are usually ionizable, and they are weakly acidic or basidic, but mostly they are bases are most common form of the drugs. So if you see here the oral marketed drugs, I got this from this review paper, yeah, the number. Almost 80% of them are ionizable, and only small number of them are neutral or always ionized. And you see from the 78 number, almost 80% of the drugs, they are mostly single base, and the rest are ampholyte and others, right? So because they are weakly base or weakly acidic, we need to talk about this acid dissociation constant. So this ionization in acidic compounds, we talk about the protonation and deprotonation. Yeah, this can be considered as one of the fast chemical reaction because it can reach the equilibrium very fast. We need to keep in mind that the larger the value of pKa, the acid dissociation constant, the weaker is the acid. Yeah, so if you have this acid, let's say HA, right? HA in aqueous solution, it can reach equilibrium and it can produce this H plus, right? And this the conjugate base. And to get the pH of it, you can just simply the logarithmic value of this concentration of H plus. This is the case for the strong acid. But what about the weak acid? In weak acid, because this equilibrium, so the reaction can shift to the left and to the right, right? So when you need to consider the pH, you need to consider the pKa. The pKa is basically just, here, I think you have learned this one before, is the ratio of concentration of the product and the reactant. So by using this Henderson-Hasselbalch equation, you can see that actually it relates the pH and pKa. And when you have the same ratio of these base conjugates and the acid, so you can get that the pH is actually the same as pKa. Let's see further here. And why are we talking about this pKa and pH? Because it's the easiest indication to check whether your compounds can be ionized easily or not in your living system. So for example, this one, diclofenac is one of the drug, right? It's the acidic drug. You see this compound here, it can be protonated, deprotonated into here, right? So when the pH less than pKa, the species usually will be protonated because the equilibrium will be shifted, right? And then when the pH is more than pKa, then the species is usually deprotonated. So here, because of that, this ionization affect the solubility. So the ionized species will have higher aqueous solubility than the neutral ones. One of the example here, the diclofenac. If you see the pH here, right? When you increase the pH, the solubility is decreased actually, right? Because when you increase the pH, the compound will be at more the neutral form, this HA form. So that's why the solubility will be decreased. And also this ionization also affect on the permeability. For example, this ionized species will have a lower passive diffusion permeability than the neutral one. So you know already this one, the phospholipid bilayer. The middle is the hydrophobic tail, right? And you have this hydrophilic head on the surface of the extracellular and the intracellular. So apparently, when you increase the pH of this acidic compound, the permeability will be decreased, right? So only the simple form of whether the drug is ionized or in the neutral form, it affect your solubility and the permeability properties of your drugs. So keep this in mind. What is the effect of the pH of the environment to the ionization level of your drug? For example, you have these acid drugs. When you put your acid drugs in the acidic environment, more of this drug will be in the non-ionized form, right? And for the basic drugs as well, if you put the basic drugs in the basic environment, So more of this will be non-ionized as well. So what happened here? You have acid drug. I have this little figure here. You have the acid drug. When you put it in the acid environment, it becomes non-ionized, right? Because when you increase the pH, you decrease the pH into this more acid environment, you will have more concentration of this H+, right? So the equilibrium will be shifted to the left. So that's why you will have more non-ionized. But when you put in the basic pH environment, basic pH, it means you have less of this hydrogen species. So the equilibrium will be shifted to the right side. So you will have more ionized species. So what is the implication of this? So it can determine whether your drug or your molecule can pass through the cell membrane passively. So here, the ionized molecule you already mentioned before, it has difficulty to pass through the cell membrane. So like in your body, it has difficulty to be absorbed by your vessel, for example, while the non-ionized, the more neutral one, is easier to pass through the cell membrane, is easier to be absorbed, right? Because the ionized molecule will be more hydrophilic, while the non-ionized will be more lipophilic. So let's play a little game here. So you have this acid drug, as I mentioned before. The acid drug, if you put in acidic environment, it will be more non-ionized, right? It has more ratio of the non-ionized species. So you have here, for example, you put this acid drug in pH about 2, you see it's more non-ionized portion, right? You increase your pH a little bit to the pH 6. The portion of the ionized molecule will be more, right? And you increase further, the ionized species will be even more and more. So from this, can you estimate what is the pKa for this drug? You have these 4 options, right? What is the pKa for this drug? So you have been experimenting, you have these acid drugs, you put in different environmental, different pH. Then you want to estimate the pKa for the drug. Maybe I go back here. So you can estimate the pKa of the drugs when the species of this ionized and the non-ionized is equal. So you can tell, oh, the pH of the environment will be the pKa of the drug. Keep that in mind, and then I think you can answer this. Then in this situation, what is the pKa, the estimate pKa for this drug? Any opinion from any one of you?
Can I answer?
Yeah, sure. Please go ahead. Oh, wait, let me check. Raihan, is it?
Yes, that's correct.
Yes.
In my opinion, I think it's somewhere around 5.5 to 5.9, so maybe 5.7. That's in my opinion.
Okay. Why you think so?
Because in the pH 6, I can still see that there's still a little bit more non-ionized molecules there, just a little bit. So basically, I think the pKa must be somewhere around there. It couldn't be higher and it couldn't be too far lower.
Okay, good. Good, thank you, Raihan. Anybody else wants to give an answer? No? Yeah, actually you're correct, but I make it more simple, about 6. So it's about 5.7, 5.9. Yeah, correct. It's easier to see because you know that when the pKa will be similar to pH, when you see the portion of the ratio of the non-ionized and the ionized will be about the same, right? About equal. So from here you can tell, oh, the pKa is about 6. Good. Any questions so far? I hope you can follow. All right, let's continue then. So from that background information, so we can know when you are given the nature of the drug, whether the drug is acidic or basic, and then let's say you already know the pKa from the calculation, the pKa of the drugs, and then you know the environmental pH of the drug is put into, then you can predict. You can predict whether the majority of the drug will be in ionized or the non-ionized form, right? And then you can also predict whether the drug is more hydrophilic or lipophilic form. And you can also predict whether the drug can pass through membrane. For example, if you administer the drug through the oral by mouth, or whether the drug can be dissolved nicely in the aqueous environment. So you can design the drug for it to be administered through the IM, intramuscular, or the subcutaneous. So let's play around again. So I give you this acidic drug. Keep that in mind, right? You have, when you are given the nature of the drug, the pKa, and the environmental, that the pH that the drug is put into, you can predict these things. Let's try and play some prediction. You have this acidic drug, this weakly acid drug. It's known the pKa is about 7. So you need to predict whether this drug candidate can be easily absorbed by the stomach, the intestine, and whether it can be absorbed well by the SQ injection. SQ is the subcutaneous, yeah? It's through the skin. Can every one of you do your own calculation, and then we can discuss. All right, you have this acidic drug, you already know. The pKa is like that. What to do? The easiest way to first do is you need to estimate whether the drug, your drug in this condition, for example, in the stomach, right? The pH is very low, about 2. So it's about here. When you put the drug in the stomach, pH 2, whether the drug will be more in the non-ionized or ionized form.
Is it non-ionized?
Hello?
Yes,
Yes.
Doctor.
Mhm.
Is it non-ionized?
Non-ionized. Okay. Why is that so?
Because this drug is a weak acidic, right? With a pKa of 7. Then because in the stomach the pH is 2, then the acidic is same with the pKa of the acidic drug. Then it will become non-ionized drug when it comes in contact with the stomach, pH of the stomach.
Good. Yes. So it will be more in the non-ionized form, right? Because the equilibrium can shift from the left to the right, to the non-ionized to the ionized. But when it's put into the pH environment that is less than the pKa, it will be favor the non-ionized form. So now you know your drug mostly in the non-ionized form. In this case, whether the drug can be easily absorbed in the stomach or not. What do you think?
It'll
Yes.
be easily.
Oops, sorry.
Sorry.
Yes, go ahead. Sorry, yeah. Again?
Yes, it can really absorb in the stomach.
Yes, because it's the non-ionized form, right?
Yes.
Good. Not is more the non-ionized, and then yes, it can be easily absorbed by the tummy, your tummy, right? And then how about in this situation, when you put your drug in the pH about 6, is the pH of our intestine. What do you think? Whether it can be easily absorbed or not? Anybody?
It's not as easily absorbed as in the stomach, but there's a possibility about just over half of it maybe would be absorbed.
Okay. Yeah. It's still some of them in the non-ionized form, majority of them, is not that easy as in the stomach situation, but still it's less than your pKa. So still some of them will be absorbed easily by the intestine, right? Now, next question. How about with the SQ injection, the subcutaneous? Usually in this situation, you need to make sure that your drugs is dissolved well in your blood, in the blood system, in the aqueous environment. So what do you think? The pH of the environment is about 7.4. Is the pH of your blood, right? What do you think? Whether it can be easily absorbed and dissolved if you use this drug for the SQ injection? What do you think?
Doctor, is it
Yes.
because the drug is the pK of 7, but the pH for the SQ injection is 7, like around 7.4, like that. So it means the drug is become ionized, then it will very hard to absorb.
Is it very, again?
Hard to absorb.
Hard to absorb. Okay. Actually, yes, it's very hard to be absorbed by the your cell barrier, your cell membrane. But since this is by the SQ injection, so it will be easy to be absorbed by the system, the blood system. Because in the blood system for the IV injection, for the SQ injection, your drugs needs to be dissolved well in the aqueous environment. So it means that your drug need to be ionized. So it's actually yes. Good. Anybody has questions so far?
Doctor,
Yes.
like if there is a bit difference in pH, like maybe like 7.1, is it effects more like on the pK, like because this is on 7.4, because the pK of the acidic drug is like pK is 7, there is a slight difference. It's not a big gap difference, so is it like give a big effect on the absorption and the ionization?
Correct. Actually, the difference is a very, very small difference, right? So if I were the chemist, I would modify the drugs so that I will have the pKa less than 7. Because let's say I want to design this drug to be for the oral drugs administration, right? So I want the drug to be easily absorbed by this stomach and intestine, right? So this is also okay. But if I want to design the drug for the IV injection, so the IV injection, the blood is 7.4. I know if the pKa is 7, it's small difference. I don't know how much of the species will be the ionized form, as you mentioned, right? So as for the chemist point of view, you need to modify the drugs a little bit so that the difference in the pH and the pKa will be more lah. Yeah, you're correct.
Then how about like if the environment of that things is same, like like exactly same with the pK of the drug, like this drug, is there any because it's like it can be either really absorbed or absorbed because it's either not ionized or ionized.
Correct. Yeah. So in that case, you need to play around with the dose because you know that this drug will be like say half, right? Half ionized, half non-ionized. So the number of drugs that can be absorbed is less than what you expected. So it means that you need to give more concentration of the drug. Then you need to play around with the dose. That's one way to do it. Yeah.
Okay. Thank you, doctor.
Good question. Anybody else have questions? All right, if not, let's play around one more time. It's the similar, but it's the basic drug. Previously, we have this acidic drug. Now we have the basic drug. It has the same pKa of 7. Please predict if the drug can be easily absorbed in the stomach or intestine and whether the drug can be dissolved. I need to rephrase this actually. Can be dissolved well by the SQ injection. Anybody? Okay, I give you some time to think and then we can discuss.
Doctor, can I answer?
Yes, sure. Adam, is it?
Yes. So In the stomach, since it's at a pH of 2 with a pKa of 7, since it's a basic drug, it will be highly ionized at a pH of 1, therefore meaning that it will have very little absorption in the stomach. Whilst in the intestines, the difference is still lower, so it'll still be ionized in the intestines, but the rate of absorption will be higher than in the stomach, but not as easily absorbed. Whilst in the SQ injection, since it's higher than that, there will be more non-ionized, which means that it'll be less soluble in the bloodstream than the previous drug.
Yes, good. I have the same opinion as you. So you're correct, because it's the basic drug, right? It's the opposite of the acidic drug. When you put into the situation or the environment with the lower pH than the pKa, it will be more in the ionized form. It's the opposite of your acidic drug, right? It's more ionized, it's more difficult to be absorbed by the cell membrane in your stomach and in your intestine. So correct, its ionized form is not easy to be absorbed, right? And how about for the SQ injection or IV injection? You're correct, because it's slightly more than 7. So more than 50% of it will be the non-ionized form, and it's more difficult for it to be dissolved nicely for the SQ injection. Nice. So what you can take from this? So you have this drug with the same pKa, but the nature of drug is different. One is acidic, weak acid, and one is the weak base. It has totally different profile in your body. So you can design such drugs to be suitable for whether it's for the oral administration or for this IV administration. Yeah. All right. I have another game here. Okay, let's play with this penicillin. It's the same thing actually. Penicillin is actually an acidic drug with a very low pKa. The value is about 2.7, and you can predict the drug, whether it can be readily absorbed or in this location, and explain why. So actually this is for your kind of assignment, small assignment. So maybe you can write it down, or you can take a picture of it, and then you can submit to Dr. Bayu, yeah, the organizer of this lecture. Okay, I give you some time to write it down or to take a picture of it. Yeah, I don't think I have the answer on this slide. Yeah. Let me just check a little, stop sharing a little bit. I want to double check. Yeah, correct. I just want to confirm whether this is really for the assignment or for our practice here. All right. Can I move forward? All of you has written enough?
I have. Yes.
Okay. Okay. And the rest? You can just give the thumbs up. You know, you have this emoticon, right? If you have any difficulty, just let me know, okay? Okay, good. I see some of the thumbs from you. Okay, this assignment you can submit later to Dr. Bayu, yeah? Okay, let's move. So we have talked about the ionization and how it affect the lipophilicity, it affect the permeability, it affect the absorption of the drug. Now we move to the second parameters. It's called the lipophilicity. Yeah, the lipophilicity it represents the affinity of the molecule or a moiety of a lipophilic environment. So it actually comes from the Greek word. Its literal meaning is fat friendly, or we call it the fat loving. So it means that the compound likes to be in the lipid environment, the fat environment, right? And apparently, this lipophilicity has major effect on many pharmacokinetics and the toxicity properties, as well as the pharmacological activities. So this lipophilicity is not easy actually to be measured in the living system, because it's very hard actually to measure how much of the drugs can pass through the cell membrane in the cell system. Yeah. So what we can do is that we make this model of this octanol and water. So we call it this the partition coefficient, or we call it the log P letter. Log P is the common estimate of the lipophilicity, because it has such impact on the drug uptake and metabolism for it to be able to pass through the cell membrane here. Okay, so let's talk about this partition coefficient, or the log P. It's the intrinsic structural property of a molecule. So basically it's the ratio. The log P is the ratio of the concentration of the neutral species in the two phases, in the lipophilic and the hydrophilic phases. It's often used for the structure property relationship analysis when you do the reformulation of the drug optimization. Yeah. So as I mentioned before, the model for the biological membrane, because it's the partition coefficient is very difficult to measure in the living system, in the native environment. It's not easy to measure. So you have this octanol and water to mimic the cell membrane. Let me just go back a little bit. This is your membrane, right? You need to able pass through this cell membrane. Where's my pointer? Here. So inside here is the hydrophobic tail, and the outside is the hydrophilic head. So you mimic that situation by using the octanol and the water. So basically what you can do here, you put your drugs into the mixture of this octanol and water. It's not really a mixture because they cannot really mix, right? But you put here and then you can shake well, kind of when you do the extraction, right? When you do it in the lab, the extraction, then you can see how much portion or how is the ratio of your drugs or your molecule that are move or resides in the octanol environment and how much of them are in the water environment. So from here, you can measure the log P as the logarithmic formula or the logarithmic value of the concentration in the octanol and the concentration in the water. That's the log P. So we have log P, we also have this log D. Maybe you are more familiar with the log P, but for the in vivo experiment, we usually use the log D. It also describes the lipophilicity, but now we know that the drug is not only in the neutral form, not only in the non-ionized form in your body, right, in your system. It may be ionized, it may be non-ionized, and most of the time it will have some portion of the non-ionized and the ionized form. So you need to put that into consideration, because in the log P, you can only measure the neutral species, right? But in the real situation, you may have the charged species. So that's why you need to put that into consideration. So the log D is the appropriate descriptor. We call it descriptor for the properties, for the lipophilicity of the ionizable compounds. Because some compounds like drugs, they are ionizable, right? As we see the earlier slides, majority of the drug molecules are ionizable. So, and because it accounts for the pH dependence of a molecule in the aqueous situation. Now you already learned, like if you put your drug in your stomach, in the blood, in the different part of your body, the ratio of the ionized and non-ionized will be different. So that's why you need to put that in consideration, and then you need to calculate the log D. It combines the lipophilicity and the log P and the ionizability of the pKa, right? So because the log D is pH dependent, it actually might be better, the predictor for the in vivo properties. So this is some of the examples of the different number of log D and log P, because log D is the pH dependent. So sometimes the value of the log D is not always equal to log P. Sometimes they're different. For example, here you see the ethanol, you see, the log P is about 0.8, but the log D at the specific pH in the blood environment 7.4 is minus 0.2, right? And let's take another example of here, rifamycin. The log P is about 3.7. It means that it's slightly have this lipophilicity, but when you calculate the log D at the 7.4, the log D is very low, 0.9. So it means that the lipophilicity property is changed if you put the drugs in this blood environment. But how about this? You see this another example of atazanavir, this drug. You see the log P is about 4, but when you calculate the log D in the blood environment is also 4. So that's it may not always go to equal number of the log D and log P, because some of the drugs, the ionizable drugs, it is pH dependent. All right, that's about the lipophilicity. Any questions before I move to solubility?
Doctor,
Yes, please. But how do I address you? Is Hezron, is it?
yeah, Hezron.
Hezron, okay. Yes,
Yeah.
go ahead.
Based on the log P, because we use the ethanol and water to predict, is it?
Octanol.
Yeah. Is there any like backup solvent, either like other than octanol or the water? Maybe some other solvents that can be replace these things.
Yeah, actually, you can use other hydrophobic, not hydrophobic, yeah, more hydrophobic solvents you can use. The point is that you need to make sure that the hydrophobicity of that other solvent is really has big difference than the water, so they will not be mixed together. Why we choose octanol? Because the polarity is much different and it's easier to get. But of course, you can use other solvents. But the number, the calculated log P might be different. So that's why the consensus is that the octanol you can use. So when someone do the log P experiment and another one in different part of the world also doing the log P experiment, what we keep in our mind, we use the same solvent, so we can compare our results, right? But if you use different type of solvent, you need to indicate that one, because the number of the log P from your experiment might be different.
Okay, understood, doctor. Thank
Yeah.
you.
You're welcome. Any other question? All good? All right, let's move then. Let's go to solubility. We have discussed ionization, lipophilicity, now the solubility. What is solubility actually? Solubility is the maximum dissolved concentration of some compounds under a given solution condition. So the number of solubility, it depends on the solution of the solvent that you use. It is actually a determinant of the intestinal absorption and oral bioavailability, because we're talking about drug, right? So we're talking about what is the implication of the drug and when you administer the drug to your body. So 70% of all cells in the pharmaceutical industry is actually for oral delivery. Why you think so? Is the data from Novartis 2018. Why you think 70%, the majority of the drug that were designed, they are designed for the oral delivery? Anybody wants to give opinion? Because there are different route for the drug administration, right? You can use oral, nasal, what else? Injection, anybody, anything. But why you think the oral delivery is more preferred? Anybody?
Can I offer an opinion?
Yeah, sure. Raihan.
Well, this is just from a business standpoint, but basically, if you create more drugs that can be, more effective through oral delivery, you are bound to sell more. Because if you were selling mostly the one that has to be you know, needs a prescription from the doctors and whatnot, you probably will end up selling less instead.
Okay, good. Anybody has other opinions? Thank you, Ryan.
Is it because it's cheaper?
It's cheaper as well. Yes. What else?
Is because from oral ingestion, it dissolves the easiest.
It's easiest? Yes.
Yeah.
Yeah. For the patient, right? You mean?
Yes.
Yes.
So it's easier for it to come
Correct.
into the body instead of
Correct.
injection.
Yeah. So all that's the opinions that you mentioned, all correct. So for the point of view of the patient, actually this is the non-invasive, right? If you compare with the IV injection, it's a very invasive. You need to prepare the syringe, you need to get yourself nick, right? It's not very convenient. So that's why the pharmaceutical industry, when they design a drug like it depends on the target location on your body, right? But majority, if it's possible, they will design for the oral delivery because it's more preferred for the patient as well. And for the storage also is easier. Usually for the IV injection is in the liquid, and then you need to have the specific condition for the storage. But for the oral delivery, like the tablet, right? You just keep it in room temperature. It has a good stability and everything. It's like that. So yeah, all cells are for the oral delivery. Oops. Okay, wait, it's not working. Okay, working now. Okay, now why is it important, the solubility? So what if you have a drug with low solubility? What problems it may occur? So you have, first you have poor absorption, right? Because the solubility, if it cannot dissolve in your system, it cannot be absorbed well. So you have poor absorption and you have the poor bioavailability after the oral dosing. And of course, if it's for the IV injection, the low solubility, you will have insufficient solubility for IV dosing. Because let's say your drug is in a solid form and it's intended for the IV injection. You need to dissolve first in some solvent, right? If it has low solubility and then it cannot mix well, right? So it has the insufficient solubility. And then it has low activity values from the bioassays because you need to do the bioassays during the drug developments. And of course, when your drug cannot be dissolved well, it will give you inconsistent and the non-robust assay readouts. And also because it cannot dissolve well, so you will need high dose administration to the patient, right? And all of this, it can lead to development challenges like the expensive formulation, the increased development time, etc. So how is the effect of the solubility in your body for this case? So the solubility, permeability, and metabolic stability can affect this oral absorption following the oral dosing. Because first thing first, when you take some drugs, these drugs in the tablet, right? The tablet, it needs to be disintegrated to your system. First, it can be disintegrated in your mouth, or maybe mostly it will be disintegrated in your stomach. Wait, I lost my pointer. Where is it? Never mind. It will be disintegrated in your stomach. Then after that, it will be dissolution in your system. And then dissolution will depend highly on solubility. After it's dissolved in your system, then it can pass through your cell membrane. And then this is what we call the permeability. And then after that, it can go to the metabolism and the systemic circulation for it to reach the target of the drug in the body. So we talk about the solubility, but how does the drug solubility looks like actually? Let's see some example here, yeah? So I have the comparison of how much things or stuff can be dissolved in 1 liter of water. I have this sugar. Wait, I lost my, okay. I lost my pointer. Give me a moment. Okay, there you go. I have sugar here. Everyone very familiar with sugar. Yeah. What is the solubility of sugar? Apparently, you can dissolve up to 2 kilograms of sugar in 1 liter of water. It's a very, very highly soluble. And how about the salt? Salt also easy to be dissolved, right? But the solubility is less than the sugar. You can dissolve up to about 350 grams of the salt into 1 liter of water. You see the difference of sugar and salt is already, how much different? Huh? About 5 times, about more than that, 5-6 times difference, yeah, already. And how about sand? We can say sand is actually not very soluble in water, right? Because have you ever seen somebody dissolving the sand in water? No, right? So visually, sand is not soluble in water, but it has some level of solubility in water. It has 10 milligram per liter. So 10 milligram is about 50 grams of sand. So still, it has the number of solubility in water, 10 milligrams per liter. How about this drug, Dactarin, miconazole, is highly, highly insoluble. It only has like 1.4 milligram per liter. Then you can compare this to the sand. Sand that is visually insoluble, but it has some degree of solubility of 10 milligram per liter. But this drug is even lower, right? Like almost more than 5 times lower. And how about this drug, Corderone? Is even lower. Like if you compare, it's thousands of 1,000 times lower, right? So when I give you this number, like, oh, the solubility is about 1 milligram per liter, but we cannot really visualize the solubility, right? But when you compare now with the sand and the sugar, now you can kind of imagine how difficult it is to be dissolved in the aqueous environment like your blood system. So how is the effect of solubility on oral bioavailability? For example, this is the development of one drug, is another HIV drug called Indinavir. So during the development process, this is the drug candidate. It has the good IC50. IC50 is when to give you the number of the efficacy of the drug, right? It has a very good number of IC50 of 0.9 nanomolar. Right? But what is the problem? The problem is apparently this drug is very insoluble. Because this is insoluble in the aqueous environment, so it has no oral bioavailability because it cannot be disintegrated in your body system. So this candidate has a problem. It has a very good potency in vitro, but is inactive in vivo just because of this poor solubility. So what we can do? So we know the problem is, this drug is cannot be dissolved well. It has a poor solubility. Then you need to increase that solubility. How? One way to do it, you learned before that the ionized form is more soluble, right? So now you can find the functional group that is not very critical for the interaction of the target. Then you can change that into the more ionizable functional group. So in this case, they change here. If you can see my pointer, yeah, here, change to here. So in the system, it will be ionized. And what happened? It will be more soluble. And then you can check in vitro and in vivo. Apparently, after you modify this compound, the IC50 is still good. You have about 0.4 nanomolar. So it means that the efficacy of this drug, of this compound, is still good. You can maintain that efficacy, but it has a very different effect on the oral bioavailability. Previously, it has no oral bioavailability because it's very insoluble, but only because you change this functional group into the more ionizable group, it has a 60% oral bioavailability in human. And then this drug can lead to, it can pass through the phase 1 to 3 clinical target. And then now it's been targeted as the HIV drug. So that's how the solubility can greatly affect the oral bioavailability. And now what we can do to adjust the solubility. Some of the examples are here. You can adjust the physical environment of your drug. For example, you can use different solvent, right? Instead of water, in general, to increase solubility, in general can increase solubility, you can use different solvent. For example, you can use polyethylene glycol, PEG, sorbitol, ethanol. It can increase up to 10,000-fold of your solubility, right? Also, you can increase the temperature. Say you increase 15 degrees Celsius of the temperature, it can lead you to about 2-fold of the solubility. You have better solubility by increasing the temperature. And also you can adjust your pH. So theoretically, when you adjust like 1 unit of pH, it can give you a better 10-fold of solubility. Yeah. And also you can change the form of the solid state of your drug. For example, you can change the crystal structure. Let's see here. You have this crystalline and you have this amorphous. Crystalline is like a very well arranged, is very nicely structured, all right? But sadly, this crystalline has a, oh, I put it wrongly. It should be the other way around. It has lower solubility. It has better stability because the interaction between the atoms is very strong. It has very good stability, but it leads to the lower stability. But when you change the crystal structure into this more random amorphous structure, so it will lead you to the more solute and solute interaction. And what is the result? The result is it will have a better solubility, but it leads you to less stability. I need to, this one should be the other way around, yeah? That. And this is the example of solid state adjustment, like for the formation of the salt. Let's say you have these drugs, para-aminosalicylic drugs. This is the original structure of this drug. You see here, it has this acid, right? COOH, right? The carboxyl. It's the free acid. Apparently, this drug has not a very good solubility. It's only 1.6 milligram per ml, right? But then you know now, when you make a salt formation of this acid form, you may increase the solubility. So this is the example. When they change this acid into, change the H with the K, the solubility increased to 100 milligram per ml. And when you change this H with the sodium, Na, it increased drastically to 500 milligram per ml, right? This is how you can increase the solubility. The number 4 is the adjusting the solid state is possible for drugs. But what do you think? Whether can you use these 3 methods, different solvent, temperature adjustment, and the pH adjustment to your drugs if you want to increase your drug solubility in your body? Do you think this is possible to do?
No, I don't think it is. I don't think it's possible. No.
Not possible, right? Because?
No. Because we're rather susceptible to
exactly
many of these things, yes.
Exactly, it's impossible for us. Do you want to swallow this solvent, or you know the body, your body temperature? You cannot adjust your body temperature. It's only like 37, the optimum condition, right? And also the pH. Whether can you adjust your pH of your blood or your tummy, intestine? No, right? So how can we do it then? You can do this solid state adjustment, or you can do structure modification. Yeah, the easier way to do is to modify your structure to improve solubility. So what you can do, as a chemist point of view, you can do some of these things. First, you can add ionizable group, just like what I showed you just now here, right? You adjust this group and changes into this more ionizable form. like you can add amine or a carboxylic group and others, or you can add more hydrogen bonding functional group. Let's say you add more hydroxyl or this amine because it can enhance the aqueous solubility. It's easier for you to do, right? Or you can add some polar group because polar is more interaction with your aqueous environment. So you can introduce ester group or you can introduce carboxylic acid groups. And also maybe you can reduce the molecular weight because we know if the drug size is bigger, it's more difficult to dissolve. So you can lower the molecular weight to increase the stability and the maintaining the metabolic stability as well. And also you can reduce the log P because reducing the log P, it means we can increase the solubility and it can lead to a higher systemic exposure. That's some of the ways that you can do for to modify the structure so you can improve the solubility of your drug candidates. Right, let's see some of the example of it. This is another example that introducing a hydroxyl group into the molecule can increase solubility because it kind of adding more hydrogen bonding, right? So this is one of the example. Is another, it's not anti-HIV, but it's anti-AIDS drugs, we call it. So during the development, we have this drug on the left-hand side of me. If you can see my pointer, but it has low solubility. It's potent, but it has low solubility. What is the implication? So it has poor bioavailability, right? Then you can do one of the strategy that we just discussed. You can add the hydroxyl group here. When you add this hydroxyl group, it doesn't change the activity of the drug, the interaction of the drug to the target, but it increases the solubility. So after you add this hydroxyl group, it has more solubility and it has more oral bioavailability. And also another example of reducing the log P. This is one of the example is the protease inhibitor is a drug to inhibit the protease. That's why we call it protease inhibitor. It's reducing the log P can increase their solubility. This is one of the example. This is the structure of the drug. So here, try to reduce the calculated log P. Here, if you see my pointer, 1, 2, 3, 4, you see from the top to the bottom, you reduce the log P from 4.6 to 2.9. What happened? Their solubility is increased from 0.001 go up to 0.07. And when you check, this is in vitro, right? In vitro test. And then you check in the in vivo system, in the animal study. And apparently it agrees with the in vitro study. You can check the Cmax is the concentration of the drugs found in the blood. So it means that the drugs already circulate to your body, right? So when you reduce the log P, you increase the solubility and you can get the drugs more in the blood because you see here from 0.1 go up to 11.4. So it means that the higher solubility, it leads to the exposure to the system of your body. That's why you can get more concentration in the blood with the better solubility of the drugs. Right, another example of improving solubility. So I mentioned before that these properties are actually interconnected. So when you try to adjust one property, it may affect the other properties. This is one of the example. Let's say I have this compound A, B, and C. You have this solubility. Compound A is very poor, 0.001. And then you try to adjust by adding this hydroxyl group. You're trying to add this carboxylic group, right? The solubility increase, go from 0.001 to 0.21. But when you see the other properties, for example, the log P is decreased, right? From 4.4 to 3.8. And about the melting point also change. So these things that you also need to keep in mind. When you want to target to adjust one property, you also need to check the other properties, right? because the structural changes can affect one or multiple properties. Any questions so far?
Can I ask something?
Sure.
So basically, considering there are so many ways to increase solubility, even just from changing the functional groups in a compound, from what I can conclude, that means you cannot just use any random functional group, can you?
You're correct. We cannot. You need to consider, because when you're designing a drug molecule, right? For example, just here, you need to know what are the functional groups that are important for the target interaction, for example. Let's say this one. So you have this functional group, right? You cannot change here because it may affect the interaction with your target. So you can add here, for example, and you can add any polar group here, hydrogen and whatever polar group that you want to add. But you need to check the overall properties. For example, you don't want to add the hydroxyl here, but you want to add the carboxylic, for example. You need to check what is the effect of the overall molecular weight. Whether if you add very big group, it will affect the whole size of your compound, right? And then how about the lipophilicity and other properties? So that's why if you add one or you change one functional group, it can affect the multiple properties. So yes, you're correct. You cannot just randomly add some functional group as you like, but you need to check how is that effect to the other properties, correct?
I see. Thank you, doctor.
No worries. Any other question before we move forward?
Doctor,
Yes, Hezron, is it?
yes, like because there's an example by reducing the log P, is it? Like when it means that when we want to reduce the log P, we need to like replace another solvents, because like different solvent have different log P. Is it? Yeah.
Okay, reducing the log P. Because if we are doing experiment in vitro, we have the flexibility, we have the freedom to use any different solvent that we like, right? But we are talking about the drug compounds. The drug compounds is designed to be active in your body. So the solvent in your body, you cannot change that because it's just there, right, in your body. So we don't really have freedom to change the solvent. What you can do to reduce the log P is just like what you do with the solubility, you change the functional group to the more, for example, you want to increase lipophilicity. So it means you need to add more hydrophobic functional group. You can add more carbon chain, for example. If you add carbon chains because it has a hydrophobic properties, so it can say increase or decrease the lipophilicity. So whatever we are talking here, the solubility and lipophilicity, we're actually talking about how we can modify the structure of the compounds, because that's the thing that we can control.
Okay, doctor, I understood. Thank you, doctor.
Thank you. Any other questions?
Doctor?
Oh, yes. Adam.
Yes, I just want to ask about, because there was an example previously where the carboxylic group was changed out, what was reacted with like a potassium or a sodium, and it showed that, yeah, here, where the and it shows that when reacting with sodium, the solubility increases 5-fold from potassium. I'm just wondering if that may be due to difference in molecular weight, or if there's some other factor that's playing into that change in solubility.
Okay, good questions. Because when you see this structure, yeah, you have this carboxylate, so you can tell this is a weak acid properties, right? Acid, and when you change this with this sodium, for example, it increases the affinity for it to be ionized. You remember when we talk about the ionization, right? We want
Yes.
to increase the solubility. We want the species of the compound to be more ionized. So you increase that solubility, right? So when you change this with the sodium, it increased the affinity of the compound to be easier to be ionized, because it's easier to be ionized, so it has better solubility. Better solubility
Yes.
in aqueous environment. That's the thing. Yeah.
Yes, yes. I got that from the free acid to the potassium and the sodium. Just wondering if there's a difference with potassium and sodium, if the difference from those is
okay.
or if that's
Maybe
just a
it's I think
example.
it's just the nature of the affinity, the intrinsic affinity of potassium and sodium.
Okay. Okay. So general. Okay.
Yeah.
All right. Thank you, Doctor.
Yeah, no worries. Any other questions? I have a few more slides to discuss. Okay, if not, let's continue. So how's the effect of the structural properties on the solubility and also the permeability, right? So we have discussed that sometimes when you adjust one property, for example, you enhance the solubility, sometimes it leads to the reducing the permeability. So it's interconnected. For example, when you want to increase the charge or the ionization capability, like we have just discussed just now, or the hydrogen bonding capacity, it will increase the solubility, but sadly, it will decrease the permeability. And we don't want that actually, right? Because we want our compound to be easily absorbed, to have a good permeability in our system, the cell membrane system. So as a chemist, we must balance these different structural features to find a good balance between solubility and permeability. Right? I want to introduce you to this term called the molecular descriptors. So whatever we talk about the solubility, lipophilicity, usually we do it, we can adjust through the experimental testing. Like you want to check one solubility, so you actually, in the lab, you check the solubility of this in different solvent and all that, and so on and so on, right? But now picture this. You want to develop one drugs, for example. You have a big number of candidates, like 10,000 of compounds, right? So if you want to adjust or change the properties, it's not really feasible for you to predict or to get to adjust the properties by doing experimentally, right? Now is the AI era, right? So we can do this by using the computational assistance. And one way is, we call it the molecular descriptors. It's the parameters of the structural properties of one compound, of some compounds. So these molecular descriptors can be defined as a mathematical representation. Yeah, on the molecules properties that are generated by some algorithm that have been developed. So these numerical values are usually used to quantitatively to estimate the physical and chemical information of the compounds. So because we think that the fastest method to evaluate whether the drug-like properties is actually met the criteria, we can apply kind of rules, quote unquote, the rules. So these rules are, we can say, is a set of guidelines for us, a chemist, to guide us for us to check what is the best structural properties of the compounds that can give you higher or better probability of being well absorbed after the oral dosing. Because doing everything experimentally in the wet lab is just too costly and is not very feasible. So we come to this era where we can do a prediction of those properties by algorithms, right? So from that, we have this Lipinski rule of 5, one of the example of the rules that you can apply when you do the designing of your drug compounds. So Lipinski rule of 5 can describe the molecular properties that are important for a drug's pharmacokinetics in the body. It also includes the ADME. So usually this one is applied when you want to design oral drugs, yeah? It is important for the drug development, because where the pharmacologically active lead compound, the drug candidates, is already optimized step by step to increase the activity and selectivity. Because you adjust dose, right? Sometimes it leads to totally different type of molecules. It's much higher molecular weight. It has more rings. It has more rotatable bonds. It has much higher lipophilicity that you don't want. So this is the rule of thumb that we can use when you design the drugs for the oral administration. There are 4 rules. For you to have a drugs that has a better probability for the oral administration, the molecular weight of your drug should be less than 500 Dalton. That's one. And the lipophilicity, the calculated log P should be less than 5. And the number of the hydrogen bond donor should be less than 5. And the hydrogen bond acceptor should be less than 10. Because if you see here, everything is like the multiple of number 5, yeah? Hence the name, the rule of 5. So you can use this as a guideline when you want to design or adjust your drugs during the formulation or the reformulation. So this is one of the example again. Oh yeah, just to refresh you, what does it mean with the hydrogen bond donor and hydrogen bond acceptor? Usually, the donor is the amine NH and hydroxyl OH, right? For the acceptor, usually it's a nitrogen or oxygen because these two has this lone pair of electron that is able to accept the hydrogen bonding, right? Hence the name, hydrogen bond acceptor. So this is some example, I think. Oh, I already get the answer, never mind. Okay, I have this doxorubicin. Doxorubicin is the anti-cancer drug, yeah? It's already marketed. You can find in the market easily. Sadly, this doxorubicin has a very low oral bioavailability. It has a good potency, but has a low oral bioavailability. So before we test this in vitro experimentally, we can actually predict whether this drug really has a low oral bioavailability or not. How? We can apply these rules, right? Molecular weight, log P, hydrogen donor and acceptor. Let's look at this. You have this structure of doxorubicin, right? You can calculate the molecular weight one by one like that, right? Long story short, I have calculated and it has 543 Dalton. And we can count how many hydrogen donor and acceptor from this compound. You can check the donor. Remember, the donor is the sum of usually NH and OH. So you can just count how many OH you see here. Here, yeah. 1, 2, wait, let me check. Can you see my pointer?
Yes, Doctor.
Okay. Okay, good. You have 1, 2, 3, 4, 5, 6, right? So you have 6 hydrogen donor. And how about the donor acceptor, the hydrogen bond acceptor? You have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12. You have 12. So do you think this has met the criteria of the Lipinski rule of 5? What do you think?
No.
No, right?
No.
It has so many violations. Even like this tree is already more than this, right? You need have more than 500, less than 500, less than 5, less than 10. So by calculating this, so you already have a sense of the estimation, right? Oh, this kind of structure, I don't think it will have a good oral availability. So this kind of set, this rule of set is not really, how to say, it's not like black and white. It's not very certain because it's just a guidelines, but it can help you to have a better, higher probability of developing a good drug, right? So one way to do it is you can just, calculate by yourself manually one by one, or if you have thousands of compounds, it's not very feasible for you to calculate manually, right? So there are many software that can help you to do that. One of the software is called MOE, Molecular Operating Environment. It's a very good software. You can do many computational chemistry there, or you can have some of the server that are available online. For example, this swissatme.ch. You can access here, and then you can calculate the Lipinski rule of 5, or you can predict some of the properties of the structural properties of your compounds. So if you have your laptop with you, or you have the you have your mobile phone, you can go to this website, swissatme.ch. When you go to this website, then you can have this window here. So you can either draw your compound, or if you already have the Smiles code is the linear form of the, is a way of writing your structure of a molecule, yeah? Or if you have the Smiles code here, you can just put, and then you can click run. When you click run, it will give you a number of properties that can, give you the prediction of how well your candidate will behave for the administration in the oral drugs. Yeah, so I think as a practice, you can later after this lecture, I think you can go to this website, and then you can draw a structure, let's say doxorubicin or any other structure, yeah? And then you can check its drug likeness properties. For example, here I use doxorubicin. It's the same structure. You can draw by yourself, or doxorubicin is already marketed everywhere, so you can find the Smiles structure easily. You can just copy the Smiles structure here, and then you can click run. I want to bring your attention here. We have calculated, I hope you see my pointer, the molecular weight from this server 543 is the same number as what we calculate manually, right? Back. We have this hydrogen, the number of hydrogen bond donor only 6, acceptor is 12, right? It's the same number of what we calculate manually. So it really saves you time if you want to predict the properties of your compounds, right? So that's what AI can do for us, yeah? Not really AI lah, but it's this digital era. Oh, any questions so far before I move to summary? I think it's time already, yeah? It's almost 2 hours. No wonder I feel so thirsty. Any questions? Or maybe
Doctor?
oh yeah, sure.
So we've obviously proposed that there is a somewhat maximum to the amount of like donors and acceptors, like roughly. Is there a minimum by any chance? Or can it just go as low as possible? Like just one acceptor, one donor, and like, I don't know, molecular weight of 100. Is that a possibility, or is that too low for a drug?
Okay, my understanding, there's no such thing as a minimum, because why this Lipinski rule of 5 comes with this number is based on the database, actually. So there are many drugs molecules, right? We have the drug structure, we have this profile and characters inside the body, and then they try to see what kind of structure that give you good oral availability, and then those database give you this number. So they can only say that if it's more than what is written here, it may not have good oral bioavailability. But the minimum number, I don't think there is any. Yeah, but for you to design say drug molecules, right? It's very difficult for you to design a very small molecules. right? Because when you design a molecule, you need a functional group to interact with the target. You have this functional group to have a good solubility, to have good lipophilicity. So those kind of, the sum of those numbers may not give you very low number of this. Yeah, I think so. You see,
Okay,
you see my
understood.
point? Yeah.
Yeah, understood. Okay, thank you, Doctor.
Yeah, you're welcome. So this is the last slide, just to conclude our lectures today. So to be a successful drug candidate, I would say, it needs high potency and activity, but also we need a very good properties. Yeah, the physicochemical properties should be considered early. Why early? Is to save you time and to because drug development process is very long, right? So you need to consider it early, especially some of the key parameters that we have discussed in this lecture, like ionization, lipophilicity, and solubility. And also the desired target property profiles, it also depends on the route of administration and the target location that you are intended to. So I think that's all for today. Any other questions before I give it back to our moderator?
Doctor,
Yes, Hezron, yes.
for the Lipinski rules of fives, because it says only for oral drugs, right? It can be used like any other forms of drugs.
Good questions. So that's why the title of this lecture is not really drug, but drug-like molecules, because this can actually be also applied for the molecules that is intended to be, for example, diagnostic probe. For example, I have experience in developing this fluorescent probe for diagnosis. For example, you want to track some protein inside the living system and all that. I can design that fluorescent probe so it can mimic the behavior of the drug, but without the potency or efficacy or interaction with the target. So because the the fluorescent probe that I need to design, it also needs to be cell permeable, needs to go through the system, right? Just like the drugs. So I can apply this system to design such a molecule for the diagnostic application, for example.
Okay, Doctor. Thank you.
You're welcome. Okay. If no question, I think I will
Dr. Samira.
Oh yes. Oh, hi.
I'm sorry,
Dr.
I have actually a very basic question. I just want to know, so when you talk about size, big drug, small drug or big drugs, it actually depends on the molecular weight, right?
Yeah.
So you have mentioned that for the oral drugs, it's normally less than 500. The molecular weight is less than 500. So basically, for the big drugs, it is used for other than oral or I don't know.
Yeah. Actually, because this number is just, as I mentioned, it's based on the database. So there must be some outlier, right? So there is one drug, I can't remember the name, but it has quite a big size. Is PSA or I can't remember the name exactly. I remember the molecular weight is about 900, almost 1,000, but it has a very good permeability and it can be applied for the oral as an oral drugs. So there is some, some outlier, but as a rule, a good rule of thumb, we can stick to this about 500 molecular weight.
Okay, so meaning that we can always say that the small drug is within the 500 molecular weight?
Yes.
All right.
Yeah.
All right. Thank you.
Thank you, Dr. Nurdini. Okay. Okay, maybe I give it back to Dr. Isna.
Thank you
Thank you,
very much,
everyone.
Bu Samira, for the informative lecture. So everyone we know today that for making a drug, we really need a comprehensive knowledge about chemistry, right? So this will make us proud as a chemist, as a chemistry student, as our expertise is valuable in every field, such as in medicinal area. Okay, before we conclude, is there any more question or something that you all want to confirm to Bu Samira? If there is no more question, I think I will conclude today's lecture. As we conclude, I want to express our gratitude for Bu Samira for sharing today's lecture about the physicochemical properties of drug molecule. And for everyone, thank you for all your participation, and we look forward to seeing you at Thursday. I think Thursday we'll meet Bu Samira again, right? Yeah, okay. Thank you, everyone. And I will give back to Hilna as the MC.
Thank you very much for our moderator, Dr. Ibu Isnani Rahmawati, SSE MSE. And thank you very much to Ibu Samira Husen Alamudi, Ph.D. for your insightful and comprehensive lecture on physicochemical properties of drug molecules. Your expertise and clear explanation have greatly enhanced for our understanding in this field. Ladies and gentlemen, now we come to the end of today's agenda. But before we conclude, we will take a moment for photo session. The photo session will be guided by Rohman. For Rohman, time is yours.
Okay, please your open camera. Okay. Oh, 1, 2, 3. Okay, again. 1, 2, 3. Okay, thank you.
Thank you very much for Rohman and all of the participants in this photo session. Before we part ways, I want to remind next lecture session about drug targets and drug target interaction with Ibu Samira Husen Alamudi, Ph.D. and will be moderated by Bapak Dewangga Ogi Bagus, SSE, MSE, Ph.D. So please make sure to join us at the same time at Thursday. Thank you very much for your kind attention. Have a great day and wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam.
Thank you, Bu
Thank you very
Samira.
much.
Thank you, everyone.
Waalaikumsalam. Thank you.
Thank you, Doctor.
Dr. Rukmini.
Thank you, everyone. Have a good day ahead.